Okay, so what I'd like to talk to you about today is mechanisms by which specific patterns of premature beats can lead to VT and VF. I'm also adding a little section on how a similar mechanism can also perhaps prevent VT. So these are the two things I'd like to talk about. First of all, is it possible to have a small number of premature beats that lead to block of the action potential wave and the normal wave, and then produce subsequently fibrillation? And then can maybe the same mechanism also in the form of premature external stimuli be used to stop spiral waves and perhaps terminate ventricular tachycardia? Okay, so this is the mechanism that I'm going to concentrate on. So what we have here is a slowly traveling wave back, which is formed through dynamics of the interaction between action potential duration and diastolic interval of a series of waves, all of which are fairly closely spaced, but some which are more closely spaced than others. So what I'm showing you here is this is an ion channel model, and I'm launching waves from the lower edge here. You can see the plane waves that propagate up. There's a slight amount of heterogeneity from left to right, which you can sort of see the wave is traveling.
What breaks the symmetry?
Right, yeah, so there's a slight, right. So I just put a little bit of heterogeneity, intrinsic heterogeneity from left to right here, which makes the wave propagate slightly differently on the left side than on the right side. And so what you can sort of see is that I've arranged the intervals between the waves so that if you notice the first wave travels very fast, the second wave travels more slowly. You can sort of see this, the wave back is traveling more slowly for that one, and the third one travels very fast. But now look how slow the fourth one travels, very, very slowly. And then the fifth wave runs into it and then produces, you get wave break sort of in the middle of the system, and then you get rotating waves. So one more time, there's a fast traveling wave. So you can see how slowly that wave back is traveling, and then you get the next wave running into it. Okay, so the hypothesis is that maybe this could be also happening just before ventricular fibrillation. So what we did is we took a couple MAPS computer model, and you've seen several examples of this in this meeting, and as with the other examples of coupled MAPS model, I'm going to have an action potential duration versus diastolic interval curve that looks like this, and also there's a CV restitution function which looks like this. So they both depend on the diastolic interval. And I'm not showing it here, but I am also using action potential duration smoothing in this to take into account the effect of electrotonic effects in the model. So and as before, I'm using space-time type diagrams to sort of get a feeling for what's happening. And then this is the basic mechanism. So these yellow things here are the action potential durations. The horizontal axis here is distance. The vertical axis is time, as we've seen before. So here, the lower edge of each of these yellow regions is the leading edge, the wave front of the action potential, and the top edges are the trailing edges. So I'm going to sort of work backwards here. So this is the kind of situation that I want to produce. So here's the last wave. It runs into here's the wave front of the last wave, and it runs into the wave back of the wave preceding it. And so what needs to happen for this to occur is for the wave back to be traveling slowly, and the next wave front to be traveling more rapidly. So how can you produce this situation? Well, here's one way of doing it. Suppose that this wave here, which you want to have the slowly traveling wave back, has a particular value of the action potential close to the launch site, but it's much longer further away from the launch site. Then you can sort of see just from the geometry of this that the wave back is going to be traveling much slower than the wave front. All right. So now the next question is, how do you produce this situation? Well, okay. So obviously, you can just take into account the APD DI restitution function. If you have a short DI here, it will lead to a shorter APD here, whereas if it's longer downstream, then it will also produce a longer APD downstream. Okay, so now how do you produce this situation? Well, one way of doing it is to have the previous wave back traveling very fast, faster than the next wave front, so that you have a shorter DI here, a longer DI there. Okay, but now here you have a faster wave back, whereas before the previous wave, this other wave here, I guess I'd say the later wave, has a slow wave back. So now in order to produce this fast wave back, you just reverse everything that you see here to produce this fast wave back. And then you can go backwards in time in the same way, alternating between fast wave backs and low wave and slow wave backs through this kind of process. So if I translate that into mathematics, this is the way it looks. For those four pictures I just showed you, that in order to produce block, you need to have the wave back traveling more slowly than the slowest wave front can travel. So you have this inequality here. And then in order to produce that situation, you need to have that the wave back is going to be traveling more slowly than the wave front by virtue of the fact that there's a gradient in the APD. There's a shorter APD close to the pacing site, a longer APD further from it, so the APD dx is positive. And then in order to produce that, you need to have also the same kind of relationship for the DIs, that you have to have a gradient in the diastolic intervals spatially, which will produce this gradient in the APDs that goes into here. And the proportionality between these two is given by the slope of the restitution function. So that's where the slope of the restitution function comes into this problem. And then in order to produce this gradient diastolic intervals, then again you need a relationship that's similar to this one, namely that you have to have the wave front and the wave back differing in velocity. So if you just iteratively put these four equations together, then you get a criterion for what can produce this block. And I won't go through the details of this, but you can sort of see that the form of this, in order to have this quantity positive here, you need to have, first of all, you need to have differences between the velocities. At least one of these pairs of velocities away, the velocity of one wave front compared to the velocity of the next wave front. And then you don't necessarily have to have, but it's nice to have amplification of this difference through the slope of the restitution functions being greater than 1, which will then create an amplification factor for each one of these things here. So this is a criterion for doing this. We didn't actually use this. We just used this criterion to check some of our results. But actually, the main way of diagnosing this was to run a coupled MAPS model. And then here's an example of one of the runs where you have the last normal action potential here with a very fast traveling wave front and also a very fast traveling wave back. And now here's the first premature action potential being launched with a very short diastolic interval here. And you can sort of see that by launching it with a very short diastolic interval, then the wave front is traveling more slowly than the wave back that it's following, which creates a gradient in the diastolic interval that's larger here than it is there. So the result of that is that the next time you launch a wave, this wave back is traveling more slowly. Then if you launch this with a longer diastolic interval, then this wave front will travel more rapidly. It starts to catch up with the wave back that it's following, and so you have a larger diastolic interval here, a shorter one there. And then if you sort of repeat the process, the second short diastolic interval really produces a huge gradient in the diastolic interval, much shorter here than it is there. So short that when you look at the wave backs that follow, that this wave back is traveling very slowly. So if you launch one more wave here, it just runs into the previous wave back and it terminates. So the sort of the most common pattern that to produce this kind of this situation is short, long, short, short in terms of the diastolic intervals. So this is the pattern that we expect to produce a wave block and then subsequent fibrillation. All right, so we've tested this. This is a few years back now. We tested this in vivo in German Shepherds. The form of the experiment was that we put catheters in both the right and left ventricles. And then the action potential duration versus DI restitution function was measured. That was the first part of the experiment. Then the second part of the experiment, we sat down with the computer code, ran approximately 70,000 versions of this coupled MAPS model to predict which combinations of the four premature pacing intervals that I just showed you will produce block. And then we tried a bunch of those pacing intervals. Of course, we couldn't try 70,000 of them, but we tried some representative sample of them, both versions that were supposed to produce block and sets of intervals that were not. So here's an example from one of those experiments. So here's dog number 1. We first measured the restitution function. It looked like this. Then we did our 70,000 coupled MAPS model calculations. And then this is basically a 4-dimensional plot showing you when the model predicts that we're supposed to have block and when you're not. So the most likely place to have block is right here in this red region. The color, by the way, is the fourth dimension, and you can sort of see the other three dimensions. So this red region here corresponds to short, long, short, short. So you can sort of, this is the first premature axis, the second one, the third one, and the color is the fourth one. So there's so it corresponds to, so this location here corresponds to 1 millisecond for the first diastolic interval, 19 for the second, 1 for the third, and then the fourth one is always short. So the model predicts that short, long, short, short should produce block and VF, and in the experiment it actually does. And we also tried other kinds of intervals like short, long, long, short that doesn't produce VF. This in vivo experiment agreed with the coupled MAPS model. Here's some recordings from the experiment. In the left, we basically had two catheters. We're recording one from the right ventricle, one from the left ventricle. We're also pacing from the right ventricle with this short, long, short, short protocol. These are normal beats here. Well, actually, these are stimulated beats, but they have a long coupling interval. But now here are the four premature beats: short, long, short, short. And then you can see that if you look in the right ventricle, that this short wave propagated to the left ventricle, produced a long action potential. And then short and then long, so you have the discordance of the APD, spatial discordance that I showed you in the X versus T plots, and then the result of that was subsequently fibrillation of the heart. Here's another example from another dog. In this case, the curve looked like this. It had an S shape instead of just a simple sort of an exponential type of curve, and then that produced a very different looking prediction for what interval should produce to produce block. So in this case, the block should occur right here, and this corresponds to short, long, long, short with these diastolic intervals. And then when we actually ran the experiments with various premature beat patterns, then we got that short, long, short, short now does not produce VF, whereas it did in the previous dog. And then the pattern which is predicted by the coupled MAPS experiment, short, long, long, short, did produce VF. So in general, we got pretty good, very good agreement between this very simple coupled MAPS model and a real animal. So more recently, we've also tried to look at this in humans. So we have some data from the MATA-2 data set. These are recordings from ECG recordings from human patients from their ICDs, and they're recordings just prior to the onset of VF or other types of tachyarrhythmias in humans. So we looked at the intervals that preceded the VF or VT, and this is kind of a summary of it. It just looks like a complete mess. Let me see if I can try to disentangle it for you a little bit. So we synchronized all of these recordings with respect to the last normal beat, the last S1 beat, and you can sort of see all of those are long, and then all the crazy stuff starts after this last normal beat. And so this is the first abnormal beat. This is the intervals, the various types of intervals we saw, and then the second abnormal, third abnormal, fourth abnormal, and all of this stuff here is VT or VF. Okay, so try to look at this a little bit more carefully. I colored in red here all those traces, those cases where the S2, S3 interval is long, and you can sort of see they all have sort of a common pattern. The S2 interval, this one is always short. Then, of course, this one's long because that's how I chose them. But then the next ones are generally short. There's one exception, this one. And then after that, everything is short. So you, again, have short, long, short, short, and then continue then into VF. The one exception is this one where you have short, long, long, short. And those are the two patterns that I showed you from the canine experiments earlier. One other pattern we saw was also short, short, long, short, short. So this is where I colored all the intervals where the S3, S4 interval is long. So we have short, long, short, short here also in addition to short, long, long, short. This one we haven't studied, so I'm not sure whether the model predicts that this is supposed to produce block and VF or not. And then I should mention that most of the cases that we have are just all short. So if you just pace the heart very rapidly, you get VF, and I guess that's already well known. So you sort of see all those down here. Those are all short, 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 short. Lots of shorts throws the heart into arrhythmia.
From ECGs, are these RR intervals or are these base beats?
Sorry.
What intervals are
Oh yeah, RR intervals. Yeah.
These are RR, so
Yeah.
this is
Actually, they're different situations. So a lot of them are sinus type beats. Some of them there was already some pacing going on beforehand, and then so those were counted too. And actually, that's what the different colors were in the first plot that we tried to distinguish among those. Okay, so here's a little analysis that we did. Is we looked at the last S1, the last normal beat, and then the 4 abnormal beats, and basically just did a histogram of them. And so what you would sort of expect, since the patterns that produce VF are either short, long, short, short, or short, long, long, short, is that for the S2, S3 interval and the S3, S4 interval, you should either have short ones there or long ones there. So I mean, so you either should have short, long, short, short, or you should have short, long, long, short. And so that long right there and that long right there go together, and then short, or else short, that long, and then short, short. And of course, when you have all shorts, then that's just what's well known, that if you just pace very rapidly short, 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 that you should get VF. But what's interesting though is that there's this gap in here between the shorts and the longs. The intermediates don't give you this block. So can we sort of show that statistically? And we sort of did. Basically, first we applied the expectation maximization method to calculate sort of the most likely mean and standard deviation for a single Gaussian curve for each one of these 5 distributions. And then we use a Monte Carlo method, just use these distributions to come up with 1,000 random distributions based on these Gaussian distributions, and saw how well those fit relative to the actual experimental data. So in the case of these two, the test shows that these two are well fit by single Gaussians, as well as the last one. Those are all fit by single Gaussians. Whereas these two, where we expect to have both a short population and a long population, are not fit by single Gaussian distributions. The p-values are very small, but they are fit by a two Gaussian distribution. So this suggests that, again, that it's evidence that this mechanism perhaps is what's causing this, although of course it's not definitive. Okay, so anyway, so we have this evidence that short, long, short, short can produce VF now, and this wave-back collision, slow wave-back mechanism can produce VF. Now I'm going to talk a little bit about the possibility of using the same idea to come up with methods for preventing VT and VF. So here's the idea. So we use electrical stimuli delivered by field electrodes. So we're talking about electric field stimulation here rather than point stimulation for the defibrillation. We apply a series of multiple stimuli, low energy stimuli. So you've seen talks about this by Flavio, for example. The stimuli should be much smaller so that because if you deliver a large one, then you expect to have cardioversion. And then the new thing here is that the stimuli I'm going to deliver irregularly spaced in time, kind of in the spirit of what we were just looking at with the short, long, short, short, to produce fibrillation. So here's kind of the way I'm thinking about this, that here's your electric field. You notice that it's a field. You have a plane wave type of, or you have plane electrodes to produce kind of a uniform field here. And then this is the way I think of fibrillation in the heart. You have a series of these you have a set of rotating waves. And so the question is, can you deliver electric field shocks here in some pattern that will stop all these rotating waves? So the way we embarked on this is to just use the thousands of computer simulations, again using the coupled maps model, to find series of patterns of shocks here that will stop these waves from rotating, irrespective of where they are in their rotation. Because I mean, in the real world, you're not going to know where these things are, where these waves are in their physically in the rotation. And if these things are just displaying discordant alternans as they're rotating, you also won't know what stage of discordant alternans they're in. So here's kind of the way we tried to model this. So to model the effect of the electric field stimulus, this is actually this is an ion channel model, bi-domain ion channel model, where I just threw in a bunch of little tiny heterogeneities in the gap junctions in this two-dimensional tissue. And then what you see here are rotating action potentials. These are already spiral waves rotating in the system. Then at t equals 0 here, I applied the electric field across the system, and you get the formation of these small virtual electrodes around all of the heterogeneities in the system. And then those waves propagate away from the heterogeneities in the system, and then attempt at least to try to kill these rotating waves. But one of the assumptions that we're making here is that we're using low energy shocks, so it doesn't depolarize everything. I mean, if it depolarized everything, of course, you'd wipe out all the waves. But since we're trying to use low energy shocks here, then I'm assuming that you don't depolarize everything, and instead you have these regions that are immediately behind the existing waves that are already in the relatively refractory regime. So when you apply the low energy shock, it doesn't depolarize those. So in the coupled maps model, I'm now going to use a coupled maps model in ring geometry, and then these electric field stimuli are going to be represented by simultaneous depolarization in those regions that aren't already depolarized, and are also not in regions that are immediately following the wave. So those represent the relative refractory regions. Okay, so here's a sort of an example, in fact, which also doubles as sort of the two major mechanisms that we see that terminate these waves. So at the bottom here you can see these are the rotating waves, and this is in the case, by the way, of one single ring. So here you have a single rotating wave, and you can see that this wave has discordant alternans on it. You can tell because of the wiggliness of the wave backs, which are the green edges of the waves. Okay, then these horizontal dotted lines here are the times at which I apply the electric field stimulus, and you can sort of see here, for example, this is an example of this gap here being almost completely filled by depolarization as a result of the electric field, but not completely. So the region immediately behind this wave is assumed to be in a relative refractory region, is not depolarized. So the net effect of this is that it just basically advances the wavefront from this point here in space to this point. And now if this wave back happens to already be traveling slowly, then the wave back just runs into it and it terminates the wave. Another more common mechanism that we saw was that you have a little bit of dynamics going on. In this case, here's a wave being the space between the waves being depolarized, and now you look at what happens as a result of that. The wave does not actually run into it, but instead what you have is that you have this severe gradient in the wave back. You have what I call retrograde repolarization. And now if you look at the gradient and diastolic intervals here between this point and this point, there's like very short diastolic interval there, very long one there, which results in a very short active potential duration here, a very long one there, which produces a very slowly propagating wave back here that the wave then runs into the second time around. This mechanism was actually more common than this one. Okay, so again, we ran like a whole bunch of a couple map simulations and just did it for all different combinations of the interval between the first and second imposed stimuli and the second and third. And I guess I'll just point you to this picture here. All of these combinations of S1, S2 intervals and S2, S3 intervals kill this rotating wave no matter where it is in space, and no matter where it is in its discordant alternans progression. So there's a fair number of intervals there that will work to kill this wave. And I'll also mention that it's in contrast to something like the ATP, anti-tachycardia pacing mechanism, that most of the intervals that we found were effective here have periods which are longer than the spiral wave period. Okay, it works even better if you're talking about a flat restitution curve, in which case you don't have discordant alternans. Now you have a really huge region, a large combination of S1, S2 intervals and S2, S3 intervals, which will stop the rotating wave. Here's kind of an example of it happening in an ion channel model. I just pinned the spiral wave here because otherwise it kind of, for this particular ion channel model, it just wanders off and you can't see what's happening to it. But you can sort of see if I start off here, I have a rotating wave, and then there are two shocks applied here. The first one produces dynamics which makes the wave back travel very slowly, and then the second stimulus advances the wave front so that it runs into that slowly traveling wave back. I don't know if it's going so fast, I'm not sure whether you can really see it, but sort of see so right there, see how slow that wave back is traveling, and then the wave front runs into it. Can you see that? It's a little bit fast. Here it is happening in a bidomain model. This was the same model I showed you earlier. It has heterogeneities in here. The first shock, the first shock produces. You can see the depolarizations propagating away from these heterogeneities, and it modifies the wavefront. It basically advances the wavefront, and then the second wave right there advances the wavefront again, and then it runs into its refractory region, the refractory region of itself. So the first wave actually kills one of the waves immediately, or actually two of the waves immediately, and then the second shock kills the remaining wave. So is that reasonably clear? Okay, so there's the process that I'm talking about. We also tried this with lots of rings. So here are 4 rings, and here I'm just showing you the red regions represent the action potential. One side of it is, one edge of it is the wave front, the other edge is the wave back. Then you see that kind of those pink flashes. Those pink flashes are me applying the electric field. So here it is running again. Okay, so we have like, my version of fibrillation or tachycardia, whichever one you want to call it, since I guess you can't really distinguish in this case. Notice the rings, by the way, are different sizes. Okay, there's the first pulse, the second pulse, and then third pulse, and then the net result of the third pulse is gone. So the wave disappears. Okay, so here's kind of these are the same 4 rings again, but in this case we're looking at this axis here is distance, this axis is time. Okay, and then what corresponds to our actual time, the various frames in this movie, are I'm applying the stimulus, this pattern of 3 stimuli at different times relative to the rotation of the wave. And what you can see from each one of these frames is that depending on when I apply this pattern of 3 stimuli, the mechanism by which the waves are getting killed is different. Okay, you can see these sudden changes in morphology or topology, maybe you would call it, of these various wavefronts. But the net result from all of them is irrespective of when I apply this, none of the waves survive to t equals infinity. So the waves get killed, so killing all 4 waves at once, irrespective of when the stimuli are applied. So hopefully this is a mechanism that we can at least start with to try to come up with sort of intelligent ways of using dynamics to kill some of these rotating waves. So let me summarize the 2 parts of my talk. So the first part of the talk, let me remind you, was can we induce fibrillation using certain intervals? And when we apply the theory, we find out that short, long, short type of patterns and short, long, long, less commonly short, long, long, short patterns of premature beats will lead to wave block and presumably subsequent VF. Then when we try to do this in experiments, in vivo experiments, we find that indeed that the same patterns appear to produce VF in vivo. I haven't talked about this in this talk, but we also see this in vitro optical mapping experiments. And then I did also talk about the fact that we can see this in human ICD data. The second part of my talk had to do with termination of fibrillation. And here what I've shown you is that a single ring, for a single ring, there are lots of different sequences of 3 field stimuli that can terminate the rotating wave irrespective of where the wave is in its rotation, either physically or dynamically. We can show that this mechanism also works for a pin spiral wave with a simple ion channel model. It will probably be necessary, although I've showed you some pretty good examples that it does work, that you would need to do other things in the more general 2D case, for example, when you don't have a pin spiral wave. The mechanism also works for 2 and 4 rings, irrespective of timing, but what it doesn't necessarily work for is it doesn't necessarily work for like arbitrary combinations of rings. So what I've shown you looks pretty good, but I have to inject a note of pessimism here that because there's really a big difference between one-dimensional rings and 2-dimensional spiral waves. In particular, there's dynamical heterogeneity in spiral waves between the tip of the spiral wave and the arm of the spiral wave. The periods are different. You can also have to worry about things like meandering that will modify the period of the wave in different locations. That I think I think we have some good ideas here, but I think there are more ideas. I feel like, in particular, maybe in a statistical regime where we need to talk about these things statistically, where we're not actually killing all these waves, with a straight 3 stimulus protocol, but maybe we need to do, maybe we'll kill some fraction of them, and then we'll need to do it again, and we need to do some sort of statistical analysis of how this might work out. I think there's more ideas that are needed here, both because of the heterogeneity of spiral waves and also because the waves, contrary to what I show here, don't rotate like 10, 15 times where you can sit there and take aim at them. I mean, they tend to rotate less than once, so what do you do in that situation? Okay, so let me just acknowledge Flavio Fenton, Jeff Fox, who's sort of one of the people that pioneered these types of coupled MAPS models, and also acknowledge grant support from NIH. So this is my website where if you're interested, I talk about a lot of the other things I do other than what I talked about today. Thank you.